Okay, let's get started. Good afternoon, everyone here. Thank you for coming here, and this is the very final event of CINP 2020 for Tokyo. And my name is Hiroki Uchida from Keio University, Department of Neuropsychiatry, Tokyo, Japan. And it is a great honor and a privilege for me to chair this plenary lecture. Today, we have a special plenary speaker from the States. Professor Virginia Lee is a professor in Alzheimer's Research University of Pennsylvania Perelman School of Medicine and director, Center for Neurodegenerative Disease Research. As you know, Professor Lee has been working on biology of tau, synucleins, TDP-43, and amyloid beta precursor proteins in health and disease. She has published tons of pivotal papers in this field, and her contribution to this field is enormous. I was very much looking forward to your talk. Oh, Professor Lee, where? Oh, you're here. Okay. Could you come up to the stage and please start your lecture?
I thank all of you for sticking till the bitter end. And so today is Sunday night, and you are eager to go home after spending here for several days. So I thank you for being here. And so as you know, that I work on neurodegenerative diseases, so I'm switching gear a little bit. And instead of talking about neuropsychiatric diseases, I talk about neurodegenerative diseases. And so what are neurodegenerative? I don't have any disclosure, so this is a slide that they put in for me because I don't consult for any company. So. And so basically, many of you, even though you're psychiatrists, you know neurodegenerative diseases, you know that these diseases are caused by misfolded proteins. So basically, there's specific proteins that are misfolded. For example, in Alzheimer's disease, you know that there are extracellular beta amyloid that accumulate in Alzheimer's patient's brain. And also, the second pathology in Alzheimer's disease is neurofibrillary tangles, tau tangles. They're comprised of tau protein. And then also, as we know later, that there's frontotemporal degeneration, and one type of frontotemporal degeneration also have accumulation of tau pathology as well. And you know that in Parkinson's disease, instead of plaques and tangle, they have Lewy bodies, which are comprised of alpha-synuclein. And also that there's Huntington's disease with polyQ, but the other disease that we're very interested in is ALS and FTD, and some years ago now, in 2006, and after really a lot of hard work, we showed that the inclusions are comprised of TDP-43, and TDP-43, as you will see, and in my talk, that are distinctly very different, and from tau in neurofibrillary tangle and alpha-synuclein and in Lewy bodies. And so another concept that that was emerging in the 80s and the 90s already was the concept proposed by Dr. Heiko Brack and his colleagues, where they demonstrated that there's the stereotypical spreading of disease pathology and as a shared mechanism, and that account for neurodegenerative disease progression. And what he meant by that was that he basically took a large number of brains, and Alzheimer's brains, and people with different duration of the disease. And so what he showed was that early on in the disease, and he was able to identify neurofibrillary tangles in the locus coeruleus and also in hippocampus later on. And then eventually, the pathology spread to the frontal part of the brain and then finally to the entire brain. So basically, each of the pathologies spread in a specific way, but they're different from each other. And this is very important in terms of our understanding and how the pathology, what we call spread or transmit, and as the disease progress. And so, but another another observation that have been around, and once we know the pathology, is that one single pathology, in this case, tau pathology, can be found in many, many clinical phenotype diseases. So, for example, Alzheimer's disease, is the most common, and tauopathies, because there's so many patients with Alzheimer's disease. But you can see down here, there's just a long list of diseases, and in fact, because of the fact that so many of the other diseases have neurofibrillary tangles, particularly Alzheimer's disease and Perelman's disease. And so a lot of people at that time, this was in the 70s, and people thought that, well, maybe tau pathology is really not important because it's a reaction to other insult, like in the case with Alzheimer's, it's beta amyloid that cause neurofibrillary tangles to form, and also similarly Perelman's disease. But what later on in 2000, what people found, and actually Mike Hutton and other colleagues found that there are mutations on the tau gene that are found in this condition called frontotemporal degeneration or frontotemporal dementia with Parkinsonism linked to chromosome 17 or FTDP17. So in these families, so if you have a mutation on the tau gene, you will get the disease. So that support the fact that tau by itself can cause a disease. And we also showed that other people have shown that Pick's disease, progressive supranuclear palsy, and cortical basal degeneration, all of these are pure tauopathies in the sense that there's not much other pathology in the brain other than tau pathology. So what about alpha-synuclein pathology? Are they more unique then to specific disease or not? But you can see here that once again that alpha-synuclein pathology are found in Parkinson's disease, also Parkinson's disease with dementia or dementia with Lewy bodies, and multisystem atrophy, which is a unique synucleinopathy in the sense that the synuclein pathology are found in glial cells, not in neurons. And Alzheimer's disease remain one of the most common synucleinopathy, and that's because there's so many patients with Alzheimer's disease, and 50% of patients with Alzheimer's disease actually have Lewy body pathology that are comprised of alpha-synuclein. And TDP-43, it looked like that there are only 3 diseases, but that's not the case because in the middle here, so what we now know that FTD or frontotemporal degeneration, there are many different subtypes as classified by the distribution of the pathology and also the clinical phenotype and so on. But again, Alzheimer's disease is the most common TDP-43 proteinopathy for the same reason I just mentioned, because so many people that with Alzheimer's disease, but there are a condition that's particularly unique, which I didn't put here. Maybe I should put it as a separate entity. And these are people with Alzheimer's disease that develop TDP-43 pathology very late in their life, and usually in the late 80s or 9th decade of their life. And so basically, there's a lot of work to be done, to be studied for late. So based on everything I mentioned to you, that there are many subtype, clinical phenotype of this with the same pathology, and also that they progress and over time. So that allow us to put forward 2 separate hypotheses to account and for what we see in patients. And the first is what we call transmission hypothesis, and that is that can we demonstrate misfolded protein, misfolded neurodegenerative disease protein, and that can initiate and induce the cell-to-cell spread of pathology in mouse models. So that's one thing that we need to do, is that if it's true, then we should be able to generate mouse models. And the second is the strain hypothesis, because there's so many different clinical phenotypes. And so perhaps then each of the clinical phenotype would have different, what we call strain, and pathological strains. And strains perhaps means maybe different conformation that exists in neurodegenerative disease proteinopathies that could potentially explain the clinical phenotype. So I want to address the first hypothesis first. And so to do this, and we basically decided that we do not want to use transgenic animals because of the fact that, when you overexpress a disease protein, like for example tau and or alpha-synuclein, and that to generate an animal model to make the protein aggregate, but that's not found in humans. And in Parkinson's disease, for example, there are only 1 or 2 families that have gene duplication or triplication. The majority of the cases do not have mutation, they're sporadic. So how do you model a sporadic disease that do not have overexpression? So what we decided to do is that we did this experiment now, was done about a dozen years ago. And so what we decided to do is that we inject an alpha-synuclein preformed fibers that made from recombinant synuclein protein and directly into the brain of a non-transgenic mouse. And we decided to inject it into dorsal striatum, and that's because the dorsal striatum connects to the substantia nigra pars compacta. So we want to see whether or not we can induce synuclein pathology in the substantia nigra. And so we were very surprised when we saw what's happening. And also, we decided to use mouse alpha-synuclein and put that into the mouse instead of using human alpha-synuclein, which we were studying at the time, because obviously we're interested in human alpha-synuclein because we're studying human disease. But we learned a lesson from the prion field, and that is that there may be species barrier. So in other words, that when we inject human into mouse, they may not work as well as if we inject mouse preformed fibers into the mouse. And indeed, that's what we did. We took mouse alpha-synuclein preformed fibers, and literally, and within 30 days of injection, we were able to see some pathology accumulating in the striatum. And so basically, we marked the pathology by using antibody to a specific phosphoform of the alpha-synuclein. So in other words, that alpha-synuclein is abnormally phosphorylated in the brains of patients, and also we try to and replicate that in animal models as well. And so you can see now, at 30 days, we see some pathology, and even in the cortex. But you can see in this particular panel, what we did was that we follow after injection at 30 days, 90 days, and 180 days. You can see that this is the injection site right here. The red dots are the pathology, the cells that have Lewy body pathology in the mouse model. And so you can see that over time, there are more and more pathology accumulating in the brain of this animal. So by 180 days, you can see extensive pathology, and in the olfactory bulb, in the contralateral cortex, in the amygdala, and also in the frontal cortex. What is really very, very interesting was the fact that, in the previous animal models for Parkinson's disease, like MPTP mouse models, they never developed alpha-synuclein pathology in the substantia nigra pars compacta. But in our model, by 180 days, we were able to see these alpha-synuclein accumulation in substantia nigra pars compacta neurons that are also acquiring thioflavin. So in other words, that the pathology at this point, after 6 months, can adopt amyloid conformation, and so that we can see this. So this is really very, very interesting, very exciting. And then we also show that over time, after injection, so you can see the pathology and up here that I described, but you can see concomitantly, and looking at the substantia nigra, the tyrosine hydroxylase positive neurons, you can see that at 30 days, you see a lot of neurons that are alive. But then over time, by 6 months, 180 days, you see that there are much fewer neurons compared to the contralateral side. And as you know, in the mouse, there's no connection between the ipsilateral and the contralateral side, so the contralateral side can be used as a control for the degeneration you observe in the ipsilateral side. And concomitant with the loss of dopaminergic neurons, and over time, we actually show that the animal developed motoric impairment. So you can see here, for example, in the rotarod assay, that by about 6 months or so, the animal they fall much easier, and also they lose strength. Their legs and arms lose strength. They cannot hang on to the Y-hang assay as the animal. As the cell degenerate over time. So by 6 months, they're not very good in holding on to the loose strength and not able to hang on. But you can see here in all of these cognitive tests and open field Y maze and so on, you don't see any differences, suggesting that this is specific for motor impairment. So therefore, it's a good model for Parkinson's disease. And so what about tau? Can we do the same experiment? Can we take recombinant and tau, aggregated them, and then inject it into animals and see what happened? So as you probably know, that early days when we try to aggregate tau, people cannot aggregate tau unless they supply them with positively charged substance, like, for example, in this case, heparin. So we can aggregate tau into amyloid fiber in the presence of heparin. But using that preparation, we inject it into wild-type mouse brain. You can see that we don't see much pathology, even 24 months. And after we injected this material, we saw really occasional inclusions. So that's not going to work so well. And so a graduate student of mine, and Jing Guo at the time, and thought of the fact that maybe, injecting an heparin prepare aggregated tau fibers is not the best solution. And in fact, recently, actually not recently now, several years ago, Mark Diamond actually showed that heparin would inhibit the internalization of many of these misfolded proteins like tau or alpha-synuclein. And you can see here the heparin doesn't do anything. You inject it, you don't see any pathology. And, but when you now aggregate it in the absence of heparin, you see a few cells in the hippocampus and in the Hylas region that show some alpha-synuclein tau pathology. But then when you inject AD tau now, so this is tau isolated from Alzheimer's brain. I'll tell you a lot more about that later. And you can see that it is capable of inducing pathology in the hippocampus and also in the contralateral hippocampus as well. This is really only after 3 months of injection. But now you follow them over a longer time of injection. You can see now that the pathology spread from the hippocampus all the way down to locus coeruleus. So this is kind of following what Brock had shown before, that there's a communication between the hippocampus and locus coeruleus. And in fact, this tau pathology also mature and over time. And initially, they're just really positive phosphorylated tau, but later on they again also like the alpha-synuclein Lewy body acquire a amyloid structure, so they're detected by thioflavin S. And so you can see that later stage, this pathology, the tau pathology, are amyloid fibrils. So basically we can then map the entire distribution of the tau pathology throughout the wild-type mouse brain to see whether or not we can determine how they spread. And basically, by doing that, we demonstrated that the transmission of the tau pathology is through the neuroanatomical connectome. So in other words, that according to the connection that had been described by the Atom Brain Atlas to show that how the mouse neurons talk to each other. So the bottom here is really the connectome, the mouse connectome, according to Allen Brain Atlas. So the dorsal hippocampus is where we inject. It connects to all of this different region of the brain. So by 6 months, these are the region that are highlighted in red. What we saw that show pathology, and for example, the septal nucleus and memory area, and ventral hippocampus, and so on. But basically, not shown here is that if we wait now for another year, so about 18 months or so, what we found was that all the different regions, they're interconnected with the dorsal hippocampus and develop tau pathology. So this is really rewarding in the sense that the spreading of the pathology and is according to neuroanatomical connectome. So what about TDP? So TDP-43 is a very different protein from tau and synuclein. And tau and synuclein are very abundant protein. They're brain-specific proteins. And when you knock out either tau or synuclein, the animal don't care. It's easy to make animal model of tau and synuclein. But TDP-43, and it's a RNA binding protein. So what it does is that it regulates RNA levels. So because of the fact that they are RNA binding protein, the they're mostly localized in the nucleus. So it's difficult to overexpress TDP-43. So right away, we decided that the best way to try to address this problem with TDP-43 being a nuclear localization, the protein localized to the nucleus, is that we basically target the protein and to the cytoplasm by mutating the nuclear localization signal. Okay, and since TDP-43 pathology are really found mostly in the cytoplasm anyway, so our model is really trying to maybe bias us to develop the model so that the animal can develop inclusion in the cytoplasm. And so we decided to use an inducible model because we know that TDP-43 could be very toxic, and if we let it express when the animal was born. So we use this TED-on model. So in other words, that when we put the doxycycline, we inhibit the expression of the transgene of TDP-43. But now if we remove the regulation by removing doxycycline, so what happened is that the TTA inducer can bind and activate the transcription of TDP-43 and the protein in the cytoplasm, the expression of the protein in the cytoplasm. So by doing it this way, and what we found was that, yes, indeed, we can target TDP-43 in the cytoplasm and detected by a human-specific TDP-43 antibody. So what we're looking at here is just a distribution of human-specific TDP-43 in the cytoplasm. But if you look down here, so basically if you use both human and tau antibody that recognize both human and tau, you can see that that TDP-43, the endogenous TDP-43 is in the cytoplasm, whereas the human is the endogenous in the nucleus, the human is in the cytoplasm. However, what we found was, again, after waiting for a long period of time, we didn't really see much pathology, and we see occasional TDP-43 inclusions. And that the transient expression also is not very high, and the lifespan of the mice are not really affected. And so this is kind of disappointing because this is really not any model of TDP-43 polyneuropathy because we don't have much inclusions. And so again, we decided to use the injection and paradigm. And so what we decided to do is to inject FTLD TDP-43 brain extract. So in other words, that we take FTLD TDP brains, we make an extract enriched for TDP-43, and we inject it into this transgenic animal. And you can see that 1 month after we inject it into the hippocampus, you can see that on the ipsilateral side in the cortex, you can see phospho TDP-43 pathology accumulating in the cortex as well as in the hippocampus. And even after the 1 month, you see some pathology already is in the contralateral side of the brain. And over time, we can see progression as well. So there are more and more pathology. But what is really more important is that much like what you see in the human FTLD TDP-43 brain, we're beginning to see some astrocytes. And you can see that, for example, here, that the white matter tract, some oligodendrocyte and astrocyte in the fornix, and also in the parameter tract and white matter tract, you can now see some of the pathology emerging, much like in the human situation. So basically, what I'm trying to say is that we have captured the essence of the TDP-43 and TDP-43 pathology and in the mouse model, and I just described. So, but in summary of this part of my talk, and what I've shown you is that alpha-synuclein preformed fibrils, they initiate the conversion of endogenous alpha-synuclein and the accumulation into Lewy body and Lewy neurite inclusions in animal models. And that alpha-synuclein inclusions, they can drive the loss of substantial nigra, post-compactor dopaminergic neurons, and resulting in behavior impairment that are very reminiscent of human PD. And that AD-tau, but not synthetic tau fibro, induced neurofibrillary tangle-like pathology in wild-type mice. And that FTLD-TDP enriched lysate induce the formation and propagation of TDP-43 pathology in TDP-43 transgenic animals. Just by looking at this summary, you can clearly see that each of the misfolded disease protein behave very, very differently, even when you try to generate an animal model of, with each of these misfolded proteins. So basically, this is the take-home as represented schematically. So what I have shown you is that you have a cell that have a misfolded protein. So for this to be able to spread, and it has to somehow get out of the cell, right? So it had to somehow be able to get into, and I put neighboring cell, but it's really more like the cell that they talk to, they communicate with. And so it would then enter to the recipient cell. And then once inside the recipient cell, they can then recruit endogenous normal protein to adopt that normal conformation. So basically, this allows you many different opportunities to interfere with this cascade. So you can block the release of the misfolded protein. So if you block it, it won't spread. Or you can block the uptake. And so basically, if you block the uptake, it wouldn't infect the cells that they talk to. And or you can prevent seeding. And also, finally, you can promote degradation. And when the misfolded protein is outside of the cell. So all of these are possible scenarios and to use for therapy. So now I want to just finish the rest of my talk. I would focus on the strain hypothesis. So the idea is that do pathological strains exist in neurodegenerative disease proteinopathies that could potentially explain the clinical diversity that I shared with you earlier. And so here, just as an example, and for tauopathies, you can see Alzheimer's disease, they have certain clinical phenotype. Obviously, dementia is one of the major clinical phenotypes. But the brain regions that are affected are regions that regulate memory. And CBD or cortical basal degeneration is different. They have different clinical phenotype, akinesia, stiffness, and tremor, and so on, are the clinical phenotype. And cortical and subcortical region, and such as frontal cortex, motor cortex, are affected. And PSP, or progressive supranuclear palsy, and characterized by down-gaze palsy and rigidity, again, and subcortical regions, such as globus pallidus and subthalamic nuclei, they're the region they're affected. So what we want to do is to really see whether or not we can recapitulate these phenotypes. And to put it, yes, so basically this is, this is now the neurofibrillary tangle, and with the flame shape. And there are these cortical, what they call cortical basal body, according to the neuropathologists, and in neurons. And also Pick's body. They all have different shapes. And we want to also be able to see whether we can demonstrate the different shape in animal models. And also that PSP and CBD, but not Alzheimer's disease, they have glial tau pathology. They have oligodendrocyte, tau accumulate in oligodendrocyte, and as coil bodies. And also you can see different astrocyte, pattern of astrocyte pathology, either as astrocytic plaques, a tufted astrocyte, or astrocytic inclusion. So all of this pathology, and we like to model. And also, on top of that, there's a complexity that in CBD and PSP, and they really only affect the pathology, only comprised of 4 repeat tau, but not 3 repeat tau, where Pick's body are comprised only mostly of 3 repeat tau and not 4 repeat tau. So there's a lot of things that we need to try to do in order to mimic the tau pathology. So more simplified way of looking at what I just said to you is that if now tau in the different tauopathies exists with different conformations, so you have different conformation, and strain 1, strain 2, strain 3, different conformation. They recruit now the normal tau, and then so that they elongate into fibrils that accumulate in the cell body. So what we want to know is that are these different pathology have different cell type specificity and different brain region selectivity and different rate of progressions. And so the way we do that, these experiments, is that we take the different brain, human brain, with the different tauopathy, and then we then extract the different tauopathies from AD tau, CBD tau, and PSP tau. And they're enriched, they're not really purified. And then we inject the brain lysate into the young wild-type animal. And then we wait for different lengths of time. Okay, and look for the spread of tau aggregates in the mouse brain. And then we can compare the tau aggregates between the different tau strains to see whether or not they are different from each other and whether or not they recapitulate what we see in humans. And so this is the work done by a former graduate student, Sneha Narasimhan. And basically what she found was exactly what we thought and happened in the human brain, in the sense that in AD tau, you see, and after this is 3 months and post-injection, and into the hippocampus. And then you can see that with AD tau, you can see tau pathology in the Hylas region. But the pathology is a little bit low compared to CBD and PSP. And particularly when you look at PSP, the pathology is really extensive. For example, in PSP, you see pathology in the entorhinal cortex, neurons in the entorhinal cortex. And, but in AD, this is after 3 months, and that you don't see any tau tangle in the entorhinal cortex. What I'm saying here is that among the 3 tauopathies, PSP tau appear to be more potent and inducing tau tangle pathology in non-transgenic mice than Alzheimer's disease. And we also can demonstrate as well the PSP tau and CBD tau, but not AD tau when we inject it into non-transgenic mice and develop glial tau pathology. As I mentioned, AD tau in Alzheimer's brain, they don't have glial tau pathology, but glial tau pathology are common in CBD and PSP. And so you can see down here the example that I give you, and CBD tau injection, PSP tau injection, you can see in oligodendrocyte because it co-label with marker of oligodendrocyte. And here again, similarly for astrocyte, you can see now the injection of CBD tau and PSP tau, you can find them colocalized with GFAP and marker for astrocyte. And so basically, this recapitulated the human situation and of the strain-specific property of PSP and CBD and AD as well. So this is really nice that we can reproduce all of this in a mouse brain. And so basically, this is just to compare the distribution. So again, we look at we create a heat map of the pathology after we injected the AD tau, CBD tau, PSP tau into animals. So you can see that basically they are really similar in terms of the region in which the pathology form. You can see the green, so the gray means no pathology, the green low pathology, the red is high pathology. But you can see that looking at the color, they basically are more or less the same. But in addition to just the tau pathology you can see that I have these red dots and also red stars and also blue dots. And those represent astrocyte and oligodendrocyte. They're in CBD and PSP and not in AD. And you can see the progressive increase of the pathology at up to 6 months and then a decrease now at 9 months. So basically, the pathology, once they induce in the brain and then after a while they decrease, either due to turnover, and there are very few neuron loss here, but late stage they do have neuron loss, but it's possibly the fact that it could be turnover, the pathology could turn over in the mouse brain. So what about synucleinopathies? And so these experiments are just very difficult to do, because what we need to do is identify a good source of synuclein pathology. And so basically, if you believe it or not, Alzheimer's cases has more synuclein pathology than definitely Parkinson's cases, but also Parkinson's disease with dementia And so you can see here, we took 4 cases of Alzheimer's disease and that have primary diagnosis of Alzheimer's disease, but also have secondary diagnosis of Lewy body dementia and dementia with Lewy body as well. And also, and cases of Parkinson's dementia, Parkinson's disease dementia, we have like 3 cases of that, and also Lewy body dementia. So, and some of these cases also have concomitant AD, Alzheimer's disease as well. So there's really kind of a close relationship between Alzheimer's and Parkinson's disease in the sense that if you live long enough with Alzheimer's disease, you develop some Parkinson's and phenotype and also pathology, and vice versa. When you live long enough with Parkinson's disease, you develop dementia, and some of the dementia are consistent with Alzheimer's disease pathology. So here, just to show you that indeed they have misfolded alpha-synuclein in the AD cases and also in the PDD cases. So what we did was that we took these brains and we need to look for we need to extract enrich for Lewy bodies. So how do we do that? So the way we did it was that we basically get rid of everything else in the brain and keep the most insoluble material. And so the way we did that was that we used high salt to extract to get rid, and also high salt with triton to get rid of all the protein. And then once, this is a very important step in the extraction of Lewy bodies or extraction of brain and pathology, and that you have to get rid of the myelin, otherwise it really gives you a lot of trouble. And then finally we extract it with other detergents to get the insoluble material. And you can see here, for example, this is the Lewy body that we recover from these extraction protocol. And you can see that there's still other proteins present, but it's nonetheless, and compared to the start material, we enriched in Lewy bodies. And we also, in the preparation, have some tau contamination as well here. But as you will see later, it does not interfere with our reaction. So in other words, that this amount of tau in our preparation really does not induce neurofibrillary tangle and in these brains. And so this is just a little bit more information on the cases. And basically, there are quite a bit of pathology that you can recover from these brains. So this is the good news. So in other words, that if any of you would be interested in doing some of these experiments, and it's not an impossible task, and to be able to generate enough pathological or insoluble, sarcosyl insoluble alpha-synuclein to do your experiment. And so basically, we took the material from the AD cases and we add them to now and primary neurons, primary hippocampal neurons. These are rodent neurons, and then we treat them with this Lewy body extract. You can see pathology from the AD cases and the PDD cases, and this is at a very low concentration, and with 20 nanogram per well of AD tau. And also AD synuclein, I'm sorry, and PSP synuclein, we can see pathology and in our primary neuron. But at 20 nanogram per well, and when we added the alpha-synuclein preformed fibrils, we were not able to see much pathology at all. So what this experiment suggests is that the material that we isolated from human brain, whether or not from AD brain or PDD brain, they're much more potent than the material that we generate in a test tube, because remember, the preformed fibril is something we generate in a test tube using recombinant alpha-synuclein and aggregated them and to form Lewy body fibrils. So this is just a little bit higher magnification in the sense that the processes, some processes do stain, but the cell body are the most prominent feature of Lewy body and alpha-synuclein that induce phosphorylated alpha-synuclein pathology. And so these pathology are very similar to with human extracts. So basically, there's a dose-response relationship. So in other words, that if you add very low concentration of this extract, you see a few neurons that develop Lewy body in the cell body. But as you increase the concentration higher and higher, you see more neurons develop Lewy body in the cell body. So that's good. So there's a dose-response relationship. But on the other hand, if we now compare what you see here, okay, these are Lewy bodies, all right? But if you now compare that with preformed fibro, so we made this preformed fibro even at much higher concentration. And so basically, even up to 1 microgram per ml, you don't see many cell body inclusion. So clearly, what we have been using, and so we started using preformed fibro and from recombinant alpha-synuclein protein, and more than 10 years ago, the first time, the experiment that I showed you earlier on, the first experiment I showed you in 2012 when we showed that human wild-type alpha-synuclein fibrils can induce in animal models and alpha-synuclein pathology. And that preparation in cell culture and only give rise of these neuritic pathology, and which is very, very different from what you see and when you add in the Lewy body extract, okay? So they're totally, completely different. So this is just a quantification, and there's a lot of pathology when we add preformed fibrils, but they're mostly, the total amount is a lot, but they're mostly neuritic, they're not somatic, but whereas the AD-tau, AD-synuclein and DLB synuclein, when we put those Lewy body preparation onto the primary cell culture, we saw a lot of cell body pathology. So now, after we've done all of this type of experiment, so it's very well that we can see, a lot of cell body pathology and here, but then the amount of Lewy body that we can prepare from AD brain and PDD brain are limited, and also not everybody have access to Alzheimer brain and also brains of patients with Parkinson's disease with dementia. And so we thought of ways of maybe perhaps if we can take the seeds, what we call the initial material from AD brain and PDD brain, and if we can amplify the conformation from these seeds with alpha-synuclein, recombinant alpha-synuclein monomer. And if we can do this, then we can generate amplified Lewy body. And if we can do that, and we call this P1, if we can now take this material and redo the same step again, so in other words, take 5% of this amplified Lewy body, or we call P1 seeds, and then incubate that with alpha-synuclein monomer, and then generate what we call P2 alpha-synuclein. And so basically what we want to ask is that, well, this is P1 amplified Lewy body and P2 amplified Lewy body have the same activity as the initial seeds from AD and DLB brains. So that's the question we want to ask. And so, basically, this is the seeds. I'm sorry, this slide is mixed up. This is the seeds from all the different preparation. And there are a lot of phosphosynuclein. So basically now these are the data. So basically, if we amplify from AD brain, 2AD brain, and 3AD brain, and PDD brain, you see that after we amplified it, and you can see that the materials consistently, after day 4, day 7, day 10, or day 14, material are insoluble. So they're in the pellet fraction and in all these preparations. So in other words, that if we take a little seed, amplified, recombinant, and alpha-synuclein, we actually can generate material that are insoluble that we can pellet and by centrifugation. Okay, so that's good news. So we can do something that we can call P1. So is the P1 material biologically active? So indeed they are. So basically we can demonstrate the induction of phosphorylated alpha-synuclein pathology in these mouse hippocampal neuron treated with amplified human alpha-synuclein P1. These are 2 different cases of P1, and there's 2 cases of AD from AD1 case and AD2 case, and you can see 2 cases all show amplified
and Lewy body, and after addition of the P1 material onto the primary neurons. And also the PDD case 1 and case 2 also show Lewy body. And compare now the pathology induced by this Lewy body material than PFF. You can see PFF clearly don't behave the same way the PFF sees when you amplify, and continue to show this neuritic pathology, not cell body pathology like the genuine and Lewy body material. So this is quantification again of the P1, and P1 behave exactly like what it should do, in the sense that it induced cell body pathology, and PFF also behaved like it should do, and which did not show much cell body pathology, and instead mostly neuritic pathology. So what about P2? Can we generate P2 as well, and to show that it has activity? So we took some of the P2 material and we added into primary hippocampal neuron, and lo and behold, you can see ADP2, and they both show pathology, and PDD P2 also. This is different concentration, 100 nanogram, 200 nanogram. They both are able to induce, and the same P2 can induce Lewy body and primary neurons, and whether or not you amplify from Alzheimer cases or PDD cases. And here, basically, we want to characterize a little bit more of the phosphorylated alpha-synuclein Lewy body pathology, whether or not they're similar to what you see in Alzheimer's brain. As you know, that these pathology are all ubiquitinated and labeled with P62. And here again, we show that these inclusions that are now labeled with phosphorylated alpha-synuclein co-localized with ubiquitin, and also colocalized with P62. So suggesting that the material that we amplify are very, very similar to what you find in human postmortem brains. And so because of the fact that we amplify Lewy body from 2 different sources, from Alzheimer's brain and also from PDD brains, are they the same or are they different? So we try to use different biochemical methods to see whether we can distinguish between them. And so one of the methods that we use is to try to digest them with proteinase K. So the idea here is that, you know, the fragment that are generated when you digest with a proteinase K, which, you know, cleave and the molecule and into different fragments. If the pattern of the fragments represent the conformation of the initial material. So in other words, that if the AD Lewy body are the same as PDD Lewy body, the fragment that they generated should be very similar. And you can see here that in AD cases that the treatment with proteinase K, there are 6 different fragments. And at the same time, you can see the same 6 different fragments were also generated with, when we digest PDD cases with proteinase K, suggesting that at least by this criteria, we were not able to distinguish between AD Lewy body and PDD Lewy bodies. And they're clearly distinct from PFFs, because you look at the preformed fibrils, they generate 3 major forms, and as opposed to AD Lewy body that show 6 different fragments, and PDD and digestion also show 6 different fragments. So finally, we want to know whether or not these amplified Lewy body and from AD or from PDD brain and would work in wild-type mice. So in other words, are they capable of inducing Lewy body in wild-type mice? And these experiments were done by Norihito Yumura, who is actually here sitting in the audience in Japan, and he will talk a lot more about this story tomorrow at a different meeting in Tokyo. But this is just a little summary of what he had done. Basically, we compare the injection of amplified Lewy body and with human PFF. So basically, we use tenfold more human PFF than Lewy body. And you can see that amplified Lewy body also induces pathology very similar to human PFF. and in the sense that the region are similar in the sense where they develop pathology. But I think amplified Lewy body and have higher pathology in slightly different region at human PFF. You can see the Lewy body here in a different brain region, and either injected with amplified Lewy body or human preformed fibrils. So to summarize the last part of my talk, what I have shown you is that pathological alpha-synuclein from patient-derived Lewy bodies induce pathology that is distinct from that induced by preformed fibrils in our cultured neuron model. And furthermore, alpha-synuclein Lewy body was much more potent than PFFs and seeding alpha-synuclein aggregation in hippocampi neurons. And Lewy body aggregates can be amplified in vitro using recombinant alpha-synuclein, generating aggregates that maintain the unique pathological phenotypes of the original material from diseased brain. And amplified Lewy body induced alpha-synuclein pathology in multiple brain regions in wild-type mice, and that the spread of this pathology induced by amplified Lewy body aggregates and distinct from that of preformed fibrils. And therefore, we think that Lewy body alpha-synuclein aggregate elicit a unique cellular response that are not exhibited by fully recombinant fibrils. And so our ability to amplify Lewy body material and then providing a large quantity of this material will facilitate research in Parkinson's disease in dementia with Lewy body and for the development of therapeutics. So this is my acknowledgement slide. So, and the top two, Jahan Ara and Nick Murata, did a lot of work on the amplification of Lewy bodies and in vitro and also in cell culture. And Norihito Umura did the work on and looking at amplified Lewy body in animal models. And Sneha Narasimhan and did the work and on in vitro and AD-tau model. And Sylvia Porter did the work and on TDP-403 that I presented earlier on. And then I mentioned and Jingguo and Hangzhou and did the work and on tau as well. And Lashmi and Shangoka, I want to mention her specifically. And she's the single person that prepared all the brain extract from Alzheimer brain, from dementia with Lewy body brain. It's really a lot of hard work to generate the material so that we can use them to inject into mice and also to study the biology of this material and in vitro. So all of this study was done in collaboration with John Trojanowski, and this is our funding source. I thank you for your attention.
Professor Lee, thank you very much for a brilliant talk. And we still have a few minutes, so I think Professor Lee can take a couple of questions. If you have questions, please.
Thank you for your wonderful presentation. I'm Nobu Hattori, Juntendo University. So you mentioned about the Parkinson's disease. Maybe your risk factor for developing to dementia involving AD pathology. So I'm very interested in this cross-seeding concept, synuclein and amyloid protein. So how do you feel about this cross-seeding concept?
Yeah, so the cross-seeding concept is very interesting. So, you know, if the brain have what we call brain trash, right? So I would say that maybe when you have already a lot of brain trash and the cells are not doing well, and also age is another factor, that all of this come together to promote the development of other type of pathology. And so, and you know, so this is what we observe, and now we want to reconstruct it, and we can reconstruct it. Now we want to understand why. But, you know, I would say that the tau and synuclein has a very close relationship with each other. And they don't form homopolymers, so they don't form heteropolymers. So in other words, that in the same cell, you have tau fibrils and synuclein fibrils. And it seemed like that when you have tau fibrils, then somehow it creates an environment that promotes alpha-synuclein fibril formation. And we need to understand initiation. Right now, we don't. That's a good question, but we don't understand why. And in fact, we have done the experiment with brains, with animals that have a lot of beta amyloid. And when we inject alpha-synuclein into those animals, there are more Lewy body in those animals than wild-type animals. Again, supporting the same concept that if you have one type of pathology, it somehow promotes the formation of other kind of pathology. But we just haven't, we don't know why.
Thank you. Thank
Sorry.
you so much.
Yeah.
Yeah, thank you. My name is Brad Hayhow. I'm a clinical neuropsychiatrist from Western Australia. I was intrigued, I guess, as a clinician by the fact that your mice clear the abnormal protein, because you seem to have a wonderful model for the pathology. I've got two questions about that, really. One is, do your mice who have lost function as a result of the pathology regain function when they clear the protein?
Well, they don't really clear the protein very effectively. You would have a few, that may show less pathology. But by and large, they don't really clear it. And also that what is interesting is that you can have a mouse with a lot of neurofibrillary tangles, or basically in the mouse, you see a lot of neurofibrillary tangle, but they don't really kill neurons right away. And they eventually compromise the neuron survival by basically making them dysfunctional. And however, Lewy body or alpha-synuclein pathology can kill neurons in vitro and in vivo. And so, I forgot, I didn't answer your question, did I?
Oh, no, I think you did, but I was going to follow on and ask whether you might have a model of recovery, if you could elucidate the mechanism by which the abnormal proteins are dealt with.
We have not done the recovery, and think the way to do that would be an inducible model. But actually, we've done an experiment with TDP-43, which I didn't talk about. And so, because I talked about inducible model, but we actually did the experiment where we showed that the animal and showed degeneration and the transgenic animal with TDP-43 pathology. But if you now put back dogs to inhibit the expression of TDP-43, the animal recover. And in fact, you can do it multiple times. If you do it the second time, the response, it's actually not as sensitive. So they don't really die, for example, as much as the first time around with a novel animal. And so, yeah, we've done those experiments, and the animal do recover. And particularly with TDP-43, it's very dramatic. And if you leave them for too long, and when they're really in the end stage, you don't recover. But up to about midpoint of the disease progression, that they do recover.
Thank you.
You're welcome.
The next question will be the last. Yeah.
Okay. So
the gentleman on the back.
Me? Okay. So thank you very much. Oh, excellent talk. So you mentioned that there are several kinds of protein aggregates in terms of the cell type specificity. Some of them is introduced to neurons, but the other ones is into gliosis. But I'm just confused because I just wonder whether the types of protein aggregates introduced into gliosis can still be transmitted to other brain areas through neuronal connections, or do they stay in their local brain regions?
Yeah, yeah, that's actually, it's a very interesting question. And so the thing is that, what we have demonstrated is the strain-specific induction of pathology. So in other words, in Alzheimer, if you take Alzheimer tau, it doesn't really induce in Alzheimer's brain, okay, you don't see glial tau pathology, okay? So we can reproduce that in an animal model, all right? And we know that in CBD and PSP, and there are glial tau pathology in the human brain, and we can reproduce it and in the animal model. And we think that that's primarily due to the strain differences, the conformation. But the thing is that the induction of glial tau pathology is very interesting because the glial cells don't really express much tau, okay? And yet, under this pathological condition, there's something that allows these cells to start making tau again. Mm-hmm. And so that now they could actually form aggregates when some of the pathological tau get inside the glial cells. And it's for synuclein, it's the same thing. It's fascinating, and there are two events. You know, one is that how would glial cell turn on the synthesis of tau and alpha-synuclein and start expressing them, and then develop the pathology.
Oh. Yeah, that's interesting.
Yeah, it's very interesting. It's a lot to do. We don't understand that.
Seems to have used up our time, so we have to close this session. Thank you very much, Professor Lee, for your wonderful talk. And I'd like to thank all of you here for your participation and active discussion. And finally, I'd like to share one important thing with you. At the beginning of this lecture, I said this was the very final event of this conference, but let me correct my statement, because the very final event of this conference is the farewell party that will start at 5:30. So if you have time, please join the party. Thanks.